So good morning everyone. Today we'll start with this HRV signal analysis. In our last class we have discussed about how the ECG signals they were acquired. Okay. Now once you get the ECG signal, so this is a demo ECG signal or the representative ECG signal from one of the volunteers. Now if you see over here, this is a P wave, then this large peak, this is the QRS complex, T wave, and then over here you see there is a U wave. Okay, so in many persons you will not find that this U wave comes out, but in some persons you will find that this U wave comes. Okay, so in this case the we can see the U wave. Okay, so this is the normal ECG pattern. Now, as the name suggests, RR interval. So what is that? What do you mean by interval? Interval means the time interval, and over here RR depicts that the interval between the two R peaks. That means the distance between this R to the next consecutive R, that is taken as one of the Y component. Okay. Then say for a particular X you have Y0, or say X0 you have Y0. Then you have X1. In X1 what you will be having? You will be having the next interval between the next two R peaks. Okay, so that will be Y1. Now this X1, it is basically your X0 plus Y0. Okay, so what is this X0? X0, we basically take it as 0. Okay, when we take it as 0, suppose over here we are having uh, 1 second, or yeah, say 1 second. Okay, now what will happen in the next interval when we'll be taking this y1 value, say 1.2, then what will happen? Then we'll be having x0, which is 0, plus y0 1 is equal to 1. So your the next x value is 1, then 1.2. Okay. Similarly, if you go towards the next one, say this is 0.9. This interval is 0.9. So what would be the x value? X value would be 1, the previous instance of this thing, plus 1.2. So this comes out to be 2.2. So at 2.2 time instance, we'll be having 0.9. So this variation, if you see, this variation is due to the variation between the two consecutive R peaks. And why it is coming? This is coming because of the dynamicity of our autonomic nervous system. Okay. So if you see, we are having this thing 1, 1.2, 0.9, and we are getting some y values, and correspondingly we get some x values. Now over here, this is one of the method to consider x values. Another group of researchers, they just take whatever the intervals you get, they take say 0, 1, 2 seconds, just like that. Okay, so over there what they are assuming, they are assuming that the delta t over here is 1 for each and every intervals. Okay. However, I prefer this first method where we add up the delta r values, r interval values, to the x intervals, because that gives you an actual understanding of the time scale. Okay, but there is a disadvantage of this one. What is the disadvantage? The disadvantage of this type of method is that if you see, for each delta t in values, delta t values, that means the delta x values, So what is happening? Your delta t values changes. In the first case, if you see, it is 1. In the second case, 1.2. But if you remember what we have done in our previous classes, in the digital signal processing classes, we have considered that the delta t values, they are equal. Okay, they are equal, and it varies from 0, 1, 2, 3, 4, because we have we usually fix the sampling frequency. So this is the problem with while we are considering the second method. This problem you will not find in the this method, where you just take the time instances as 0, 1, 2, 3 as such. Okay, just so this is the problem with this first method. Your Fs, you cannot fix Fs because your delta t, it is changing randomly. So under this condition, what you do is that we plot a spline. It basically takes an average of all these values and tries to draw an average line. Okay, so we take this spline function and then we resample the data. Okay, now what this spline does, it gives us an it is basically an opportunity to interpolate data. If you see, you are having samples point at 0, 1, 2.2, you are not getting at 2, but you have to get a point at 2, because if you want to process any digital signals, its time instances should be say 0, 1, 2, 3 and so on. Okay, but in this case we do not have that what you call liberty, right? So we what this spline function does is that it goes for an interpolation function. It is basically an interpolation function. There are other interpolation functions also, but the spline is very commonly used. Okay, so after you feed the data with this function, then you go for the resampling of the signal. So this resampling can be done at a particular frequency. If I say FR, now this FR would be used as Fs. Okay, when you are resampling data at a particular frequency, that is basically what you are doing. You are artificially creating Fs. Okay, then only you are able to use this whatever the data you have got for actual purpose. Okay, any doubt over here? Anyone? No? Okay. Now, why we study heart rate variability? We have already discussed that HRV, it is a marker of parasympathetic and sympathetic activity, and it is extensively used to measure the ANS behavior. Okay, so what is the ANS behavior? The dynamicity between the parasympathetic and the sympathetic activity. Okay, from the literature we have that the frequency domain parameters, they can portray more clear picture of the ANS physiology, right? So for this purpose, what we have done is that we have taken the 5 minute of ECG data from 15 volunteers. Okay, then these 5-minute signals, they were used for HRV analysis. Okay, there are 9 frequency domain parameters such as VLF, very low frequency. VLF means very low frequency. LF means low frequency. HF means high frequency. Okay, so VLF, it starts at 0 to 0.04, then 0.04 to 0.15 hertz. Okay, then HF equals to 0.15 to 0.04 hertz. Okay, and then we have LF which is a normalized unit of LF, HF unit, HF normalized unit, then VLF percent, LF percent, HF percent, and LF by HF ratio. Okay, now if you go over here, if you see, we plot the spectral power spectral density. Okay, we plot the power spectral density with respect to the frequency. Basically, what we are doing, we are basically doing calculating the x omega square. Okay, that is called power spectral density. Okay, so if you see spectral square, okay, and that's why you are getting spectral square, and that is called power spectral density. Now you plot this data. Now according to the frequency component, what we have said, this green highlighted area, it is VLF region, very low frequency region. This blue shaded area, this is low frequency region, and this red shaded area, it is the high frequency region. And from here you calculate all these powers. The software will calculate for you. Okay, so you get all these informations from here. Okay, then you have LF normalized unit, HF normalized unit, and LF by HF ratio. Okay, now this VLF percent, it is calculated by this formula. LF normalized unit, it is calculated by this formula, and LF by HF ratio, this is being calculated by this formula. Okay, now if you see the statistical then what we did is that you have also done in the practical classes, that once you have got the features, we go for the statistical significance test, okay, of these parameters. By what? By using non-parametric Wilcoxon signed rank test. So why we have done this? If you see, in many of the cases you must have heard of t-test. Okay, the t-test, however, it is used when your data follows a normalized distribution. However, for the biological data, in most of the cases you will find that the distribution is not a normalized distribution. Hence, usually for biological data, this Wilcoxon's signed rank test, it is used for calculating the statistical significance between the two groups. Okay. Apart from this, we have used two tree-based classification algorithms, namely the classification and regression tree, CART, and random forest, RF, for calculating the significant or the important parameters which have played an important role in the classification. Okay. Now if you see from here, what we have got, that VLF power, it is not statistically significant in the Wilcoxon signed rank test. However, for the CART analysis, it was found that it is playing a significant role. Okay, the LF new values, it was significant through the Wilcoxon signed rank test, and LF by HF ratio, it has played an important role in the classification using the RF. Okay, so what we have done, we have found that these three parameters, they are statistically important parameters, and they may be further used for analysis. Okay, so if you see what we have found that LF new power, it has decreased, okay, post coffee consumption. So if you see, it is 945 and it has become 744. Okay, so it has decreased after a single dose of coffee. that indicates a reduction in the sympathetic activity. Okay. Now, the second thing what we have got VLF power, which is assessed from the short-term recording, is skeptical. Okay, hence the analysis with VLF power is to be avoided. Okay. So when you talk about this signal processing and all these things, what happens is that you have computer who will be doing the processing for you. That's very good, not a problem over there. However, you have to know the limitation of the analysis what you are doing. Okay, so the cardiological society, American Cardiological Society, they have categorized the HRV analysis into short-term and long-term analysis. The short-term, what they have said is that a minimum of 5 minutes of data is required for short-term analysis. Whereas for the long-term analysis, you need at least 24 hours of data which are acquired through Holter monitor. What is Holter monitor? Anyone? What do you mean by Holter monitor? No one? No one knows Holter monitor? Am I audible?
Yes, sir.
Yes, sir.
What? Yes, sir.
Yes, sir.
So what is Holter monitor?
Yes, sir. Don't know, sir.
So tell me that, tell something. You people are just mum like anything. So Holter monitor is a portable device, okay, which is used to record long-term ECG signals. Okay, so it would be it basically the what you call the first generation Holter monitor. They usually consisted of 3 or 4 channel Holter monitors. Okay, however, as the time progressed, you can find some 12 channel Holter monitors also. They have inbuilt memory into themselves, and the data is being recorded into that inbuilt memory. Okay, and it is able to collect data for a longer period of time. Okay, whereas the normal ECG machines, they can also record signals either in the 3 channel, 4 channel, or 12 channel, but they do not have sufficient memory to store data for a longer period. Okay, so American Cardiological Society, they have fixed, the task force has fixed that for short-term recording you have to take a minimum of 5 minutes of data, and a maximum of, and for the long-term analysis you have to use a 24 hours of data. Okay. Now, since we have used 5 minutes of data, the task force has recommended that under this condition, even if you get VLF power as the statistical important parameter, this should not be used for the analysis. Okay, so hence we rejected this parameter for any inference. Okay, then what we did is that we found that there was a significant decrease in the LF by HF ratio. Okay, so LF by HF ratio, it shows the dynamicity either towards parasympathetic or towards sympathetic activity. So over here we found that there was a decrease in the LF by HF ratio. as compared to the previous one. Now, this suggests that there is an increase in the parasympathetic activity. There is an increase in the parasympathetic activity. Okay. So, from this 1 and 2 parameters, okay, LF new power and the LF ratio analysis, we can say that there is a significant alteration in the ANS activity after we consume the caffeinated coffee. Okay. Now, next our hypothesis was variation in the ANS activity. Okay. So, why this variation in the ANS activity? So we have seen that there is a variation in the ANS activity. Okay. Now, our hypothesis is that this autonomic nervous system, it is getting innervated on the that is innervated on the heart muscles. Okay, and it is controlling the cardiac electrophysiology. Okay, so there is a high probability that the variation in the ANS activity would also lead to the changes in the cardiac physiology. Okay. Another important thing over here is to see that we have not done any artificial intelligence based machine learning analysis over here. The reason behind this is that the number of volunteers what we have got is only 15. Okay, so we have got 15 multiplied by 15. Okay, multiplied by 15 means pre and post, not exactly multiply. So pre 15 data and pre post data. So out of that, a total of, you can say, a total data set would be of 30 features. So each feature will be having 30 parameters. Okay, now this 30 is very less. As for the what do you call, as per the recommendation uh, for the analysis of the biological signals through artificial intelligence based method, you should have at least 200 number of features for each of the parameters. Okay, that means you should have, if you are doing pre and post data analysis, you should have 100 volunteers, pre and post at least 100 volunteers. And if you are doing that between two groups, that one group would be you are giving the placebo, in another group you are not giving the placebo, then you should have at least 200 volunteers who should be participating in your study. Okay, so this has to be made clear in mind. Okay, so if you are doing any artificial intelligence based analysis, you should have at least a data set where you will be having 200 what do you call, 200 number of features for each feature set. Okay, or 200 number of values for each features. Okay, then only you will be able to apply the artificial intelligence based method. Okay. Then coming to the analysis of the cardiac physiology. Okay, so uh, this part what I will be teaching, because has published this paper in Medical Hypothesis Journal. Okay, so what is his hypothesis? So his hypothesis was that the effect of coffee on ECG using morphological features, it has given mixed results. Okay, then the second is performance of statistical, sorry, performance of statistical and entropy based features on other ECG application, it has shown very good results. So his hypothesis was that if we mix these two uh, components, then we may get a better classification accuracy. Okay, so for um, testing his hypothesis, what he has done, he has taken 5 segment ECG signals. As I said in my previous um, what you call slide, that you need at least 200 numbers of values for each data set. Okay, and we have got 5 minutes of ECG signal. So another way, we cannot increase the number of volunteers, okay, which are fixed as 15. So another way to increase the number of these values, these feature values, is to take the smaller segments of a bigger signal and then extract the features of those smaller segments. Okay, so and this is a valid method, and many researchers, they apply this method when the number of volunteers, they are small. So he also did the same thing. He took 5 second ECG segments before and after 10 minutes of coffee consumption. Okay, he then extracted the features from each of the 5 segments. Okay, then he determined the significant feature using the Wilcoxon sign rank test. Then he went for classification with optimal feature selection, and the optimal feature selection was carried out by a recursive feature elimination method, and then he went for the performance evaluation. Okay, now the hypothesis consequence, which is basically the result, he found out that there are significant changes in the several statistical and entropy based feature after coffee consumption. Okay, so hypothesis we have already discussed. Just a point to be noted over here. In recent years, the statistical and entropy based features have emerged as an efficient method to extract hidden patterns from the ECG signals. Okay, these features have been successfully exploited in arrhythmia detection, noise removal, and biometric identification, and many other things. Okay. Now, if you see, a subtotal of 1260 number of ECG segments of duration 5 seconds each were extracted. So, if you see what we have done before applying this, we have artificially increased the number which was much higher than 200. Okay, which was much higher than 200. Okay, now from there, from each trial 5 seconds, he calculated 5 statistical parameters: arithmetic mean, median, variance, kurtosis, and skewness, and 34 entropy based features like log energy entropy, Shannon entropy, Rennie entropy, and Salis entropy. Okay, now these features, they can be classified into 3 categories: arithmetic mean, median, and variance. They are regarded as lower order statistical features. Then we have higher order statistical features. These are kurtosis and skewness, and we have entropy based features. Okay, now for the entropy based features, among the entropy based features, the Rennie and the Salis entropy, their order can be changed. This order Q, it can be changed, and he changed the values in the range of 0.1 to 5 at an interval of 0.3, and he calculated a number of parameters, okay, a number of entropy features. Now, from the Wilcoxon sign rank test, he found that there are 20 number of features which are significantly different, and he selected the p-value. The p-value was 0.05. Okay, so if it was below, the p-value was below 0.05, the feature set was regarded as the statistically significant. It was observed that in most of the cases there was an percentage increase in most of the significant features, okay, after the coffee consumption. Now then he plot the percentage increase in the significant features. Okay, so among all these things, if you see the median, variance and standard deviation, there was an significant increase or multifold increase in these values. So what is this? Median means it gives you an information about the not exactly average, but what is the what you call middle value of the amplitude. Okay. So if you see the middle value of the amplitude, it has increased. Okay. What is standard deviation and variance says? It says from the average how your data is varying. Okay. So if you see, when we are talking about median value, that means the average amplitude value of the ECG signal have increased post coffee consumption. Not only that, there was a huge variation in the data point as compared to the average values. This standard variance and standard deviation they suggested. Okay. Then another important thing to note is that skewness, it has significantly decreased. Okay. Now, why is that? What is skewness? Suppose you are having a normal distribution like this. Now, if you see, it has a tailing effect. Okay. So it starts at A and it ends at B. So it attains a peak. Now, if you plot this one, it attains a peak pretty quickly and it dies down at a relatively slow rate. So that is skewness. So this is called skewness because while going up it is taking lesser time and while coming down it is taking longer time. So this mismatch in the time, it is called skewness. Okay. Now if you get another value like this which dies down quickly, then the curve denoted by this blue line, it would be said that the blue curve, it has a lower skewness. That means the curve is rising quickly and it is dying down quickly. Now if you see this skewness value post coffee consumption, it decreases. That means it is rising quickly and then it is relaxing quickly. Are you getting my point? So the skewness, it is decreasing. Okay. And when we are talking about this parasympathetic system, it increases the magnitude of the contraction of the muscles. Okay. So there is an increase when it is contracting. When the magnitude is higher, the amplitude is getting higher and hence the median value, it is getting higher. And at the same time, you can see the heart muscle, it is relaxing also quickly. Okay. So these are the two important things which we can uh, what you call predict from this percentage change after coffee consumption. And if you look into the other parameters, yeah, there was some increase, but that was not much significant, not much higher as compared to the these four components which we have discussed now. Okay. Now what we have done, he went for the this artificial intelligence based analysis where he applied this recursive feature elimination method and find out the performance in each of the classification models. So over here he has used this ensemble classifier in this study. Whether he has written, no, I cannot find it over here, but he has used ensemble classifiers, yeah, in the next slide. So he has used 5 different types of ensemble classifiers. Okay. So etc, card, AdaBoost, gradient boosted tree and random forest. Okay. So why ensemble classifiers? The reason behind using an ensemble classifier is its advantage to reduce the misclassification rate of a weak classifier by introducing as well as aggregating several classifiers. So basically what it says is that while you apply this ensemble classifier, it will not train only one or two what do you call uh, artificial intelligence based models or the machine learning models, but it will generate a larger number of models, and then it will give you an average output. So from there you will be having some weak classifier, then you will be having some strong classifier, and you will be having an average value. So the what you call the ensemble classifiers, the reliability of these classifiers are much better because of this reason, because it is taking the lower information also, lower classification information also, it is taking a higher classification information also, and then it is giving you an average. Okay, it just like in a class, um, if I just take the better performing student and if I compile their marks, it will not give me the actual information about the whole class. The same goes if I just take the marks of the students who are not performing well over there also. So if I take that information over there, that would also not give. However, the average, if I take the average values, I would get a certain idea, yeah, what is the standard of this class, and I can compare with the standard of the other classes. Okay, so from there I can have an understanding how the class A and class B they are performing. Okay, so the performance can be easily predicted, and that would be much more reliable. Okay, so herein the main idea was to make a prediction of several classifiers on the original data and then make a strong prediction by combining all the predictions. Okay. So here, since we have used the optimal, determine the optimal features sets by recursive feature elimination method, if you see, um, we have found that at different levels we were having a different number of the feature sets, and that values have been mentioned over here. The first one etc, it was having 9 features. The second one cart, it was having 6 features. The third one adb, it was having only 3 important features, and likewise. Okay, and all these values have been tabulated. Now when you wait for the analysis, the accuracy of the random forest, it was the highest. Okay, it was the highest. If you see, it is having a classification accuracy of 75%, and for biological signals, a classification accuracy of more than 60% is considered to be a good classification accuracy. Okay. So hypothesis consequence, we have presumed the hypothesis that statistical and entropy based features obtained from smaller segments of ECG signals can efficiently be used to detect the short term effect of coughing, and it is supported by the our findings. The existence of several statistical and entropy based features were found through Wilcoxon's Signed test, and the second is the higher classification accuracy was achieved in differentiating the two groups of data pre and post coffee consumption. Okay. Then the analysis of cardiac physiology by wavelet based analysis we have already discussed. So in the previous case we have seen that we have only taken the time domain signal. Okay, and for the decomposition based analysis, no study has been performed on the coffee consumption. So that's why we have taken this discrete wavelet transform and the wavelet packet decomposition, and we have tried to analyze the thing. The analysis was similar, the hypothesis testing basically. 5-second ECG signals was taken. Then these signals were decomposed. This is an extra step. We have seen that the signals can be decomposed using the WPD and the DWT methods. WPD is wavelet packet decomposition, DWT is discrete wavelet transform. Okay. So then feature extraction, then you have significant feature detection, then performance evaluation and all these things, right? So we have all discussed about the discrete wavelet transform and discrete wavelet packet transform, wavelet packet decomposition techniques. So I will not be going over into details over here. So over here, the decomposition based techniques were used in the decimated forms. I have already told you what is the decimated form. If at each level you reduce the size of the signal, then it is a decimated form. And if you do not reduce it, it is your undecimated form. Okay, both have their advantages. In the undecimated one, you will be getting the full length signal at each of the nodes. Okay, whereas in the decimated form, at each of the nodes you will be getting a reduced sample size. Okay, so this is discrete wavelet transform. We have already discussed. This one he has written wrongly. So this is 90 to 180. Okay. So yeah, 90 to 180, and then each levels. These are the gray colored boxes. These are the nodes, and these signals were taken, and their features were computed. Okay. So this is how it looks like. So this is a schematic representation of a three-level DWT decomposition of a representative signal. So it looks something like this. So if you see the approximate coefficients, uh, they are mainly consisting of the ECG features. Okay. Then in the wavelet packet decomposition, as we have already discussed, it is an extension of the DWT. Okay, and unlike in DWT, WPT decomposes both approximate and detailed coefficient. So if you see, it is being uh, the approximate and detailed coefficient both are being what do you call both are being decomposed. Okay. Now from each of these nodes, we take all these signals and then we go for the um, uh, calculation of the features. Okay, so this is again the representative decomposition of a representative signal. So you can see these are there. Now if you see, most of the ECG signal parameters, they are within the lower frequency components. Okay. So in this study, what he did is that he changed the level of decomposition from 2 to 5, and the mother wavelet he used dB2, dB4, dB6 and dB8. Okay. So significant feature detection. So nine statistical and entropy based features as represented in the previous series were extracted from each node of the decomposed signal, and then he did the Wilcoxon sign rank test. Okay, and then he used these important values uh, and went for the what you call for the analysis. Okay, over here he have just showed this level 5 of decomposition results. Okay, and so over here, if you see for DB2, DB4, DB6, DB8, he has found important, yeah, good, what do you call, significant features like kurtosis, AC, and all this log energy entropy and all these things, right? And here he has shown the combining all these things. What are the important features? So if you see, most of the features they are in the 16.87 to 22.5, and they are in this 0 to 5.62 region. And why that is so? That is because the QRS complex, it is having a frequency component within 16 to 20 hertz, and mean P and T wave frequency is 3 hertz. Okay, below 3 hertz. Okay, so due to this reason, if you see in these areas, if you find out, the features were quite higher as compared to the other feature sets. Okay, so this was for the WPD and this one is for the DWT. Here also you see he got similar results. Okay, if we compare here, you see this and if you see this. Okay, so it is quite um, what do you call, understandable from here that the coffee, it is not affecting not only a particular heart muscle, but it is affecting the whole of the heart muscles. Okay, so it is not selectively affecting, what I meant to say, it is not selectively affecting any ventricle or the atria. Okay, it is affecting whole of the cardiac muscles in a similar fashion. Okay, it is not selective to any part of the cardiac muscles. Okay, so then he went for the machine learning models. These are the learning models. Now here if you see, we have just used only one this ensemble classifiers. There are others also, but mainly this RF has been used. This DT, uh, what is it? Yeah, decision tree. Yeah. So the decision tree, it is also there. And then gradient boosted tree is also there. So these three trees were here. But our main idea was to analyze this RF classifier as compared to the other classifiers. And if you see over here, this RF classifier, it is not performing well over here. In fact, it is the gradient boosted tree which has performed well, and it has been in the case in all of the studies. And if you see in level 2, level 3, this thing, your classification accuracy is more than 72%. Okay, both in the WPD and the DWT region. Okay, and lower the level, better is the classification accuracy. Okay, as you see, as we go towards higher this thing, there was a reduction in classification accuracy. Okay, so why this is so? This is so because when we are going towards the lower frequency domains, we have seen all the frequency components, they are present in the lower domain. Okay, and if you go towards look into this features, level 2 is this one. In the level 2, you see, you're having a completely denoised signal. The signal looks like a denoised signal. Okay, so that means the signal what we are getting uh, a denoised signal, and which is devoid of most of the noises. Okay, and it contains all the feature components. If you see 0 to 45, it contains all the frequency components over there. And as you are trying to go towards the higher frequency components, you see you are basically distributing the frequencies components. That might be one of the reason why you are getting good accuracies at lower levels. Okay, so I just said that the alteration in the cardiac physiology is also evident by the higher classification accuracy, which is being achieved by these machine learning models. So now I come to the understanding the effect of coffee on brain cognitive behavior. Okay. In this study, we have used steady-state visual evoked potentials, SSVEP signals, which basically elicits or what you call excites the parieto-occipital region of the cortex. Okay, when a light source of 3.5 to 75 hertz, they are being flickered at a constant frequency. Okay, so why this is important? Caffeine has been found to enhance the vigilance and executive control of the visual attention. Till date, no study has been conducted which has investigated the effect of the caffeinated coffee on the SSVP response. The current study proposes a data mining based approach to study the effect of consumption of caffeinated coffee on the generation of SSVP signals. Okay. So what we have done over here, we have acquired the ECG signals. We went for the pre-processing of the signals. Then we went for the detection of the SSVP signal by canonical correlation analysis. Okay. Then we went for the feature extraction by RQA method. It is another method. Then we went for the feature reduction. Then classification. In this study, we involved 6 volunteers. Okay. This is a very tedious task, and many volunteers they back off from the study. Okay, so that's why we have a very small volunteer site. So we acquired the EEG data, and the age group of the volunteers were 18 to 22 years. Okay, we used 22 channel EEG recording machine, which is a commercially available EEG machine, and stimulus one, we gave LED flickering, which were flickering at a particular frequency in the range of 3 to 30 hertz, and stimulus 2 was the caffeinated coffee. The electrode placement was the electrode was placed in the 1020 international standard. Electrode scalp impedance was kept at lower than 20 kilohertz, and the sampling frequency was 256 hertz. Okay, from here we selected 7 frequencies, representative frequencies. Why? Because these 7 frequencies, they represent the frequency band of the EEG signal of different EEG components: delta, theta, alpha, low beta, beta, high beta, and low gamma components. Okay. So from a preliminary analysis from the brain mapping, we found that there are some changes. So before and after the consumption of the coffee, we can find that there are some changes, but we cannot get a quantitative approach from here. So what we did, we saved the data in this format. Okay, so for each of the volunteers, we got this data in this format, and they were saved in this fashion. Okay. So after the data was saved, we went for the Butterworth filtering using the 6th order Butterworth filter, and this was the bandwidth of the Butterworth filter, and then we applied a moving average filter having a window length of 40. So then we went for the SSVP detection using CCA coefficients. Okay. So I'm not going into the details of this one. So over here, what happens is that we calculated the normalized SSVP counts, and we found out that this portion, the right portion of the parieto-occipital lobe, they are being activated the most. Okay, even you see the temporal lobes, they are also getting activated with certain extent. Okay. Post coffee, we found that this P4 channel, it was not activated much. Okay, it was not activated at all, in fact. Okay, however, the other coffees, other channels, these green channels, they were activated. However, for the further analysis, what we have done, we have included this P4 channel also for a better understanding what is happening. Now, after we have done this, then we have gone for the percentage change in the SSVP activity. Now, if you see the percentage change in the SSVP activity, these frontal parietal, these PG regions, or these frontal regions, basically posterior frontal regions, they are getting much more activated. Why this is so? Because we have given visual signals, so we were expecting that the parieto-occipital lobe, it would get more activated, but the change in activity, if you see,
it is more in the frontal region. Okay, but if you look into the activity of this parieto-occipital lobe, the activity is more in the parieto-occipital lobe, but the percentage change in the SSVP activity, which is associated with the cognitive perception, it is more in the frontal lobe. This was a new finding, and no one previously has reported this kind of finding. Then we went for this activation based on the this stimuli frequency. Okay, and if you see the 30 hertz stimuli frequency, it is the most it is showing the most increased SSVP activity. Okay, so the higher frequency component, they are able to stimulate the um, uh, they are able to affect the cognitive perception the more as compared to the lower frequency components. Okay, so then we went for the RQA analysis, which is recurrence quantification based analysis, and then we performed the significant feature extraction using CART, BT, and random forest. Using the Wilcoxon's test, we could not find any significant features. Okay, then we trained the MLP in neural network for the analysis, and these are some of the features which were extracted. Now, at the end, if you look, the P4 and O2 channels, these two channels, they are being activated at majority of the frequencies. Okay, so PZ, these two channels, but their classification efficiency are not that high. So I would stick with P4 and O2. Now, if you go to the brain map over here, where is P4 and O2? The P4 and O2 over here are over here. Okay. So P4 and O2 are over here. Now, if you see in the SSVP signal component, when we are trying to analyze, the P4 was giving us a dubious result, right? But in the classification thing, if you see, it is quite high. So what does that mean? That means whatever the analysis we have done, mostly we have done in the linear domain. Okay, we have just seen whether it is present or whether it is not present. But there is a high probability that if you place the data in a non-linear dimension, which the neural network can extract or can classify, if you put it over there, then you will find, yeah, there is a possibility that there is also an excitation over there. And while we are drinking the coffee, the P4 and the O2 channels, they are being excited the most, and it covers, see, most of the frequency components. Okay, except this 10 hertz, you see, combining these two region, these two channels, we get all the frequency components, they are being excited. And if you look into the 10 hertz component, none of the channels, they give you a better result. Okay, none of the channels, it gives you a better result. Now if you go back, that means the alpha frequency band of the EEG signals, they are not at all affected by the consumption of the coffee. However, the coffee can affect all other frequency components in the P4 and O2 channels. Okay. So with this, um, I come to the summary of this section. Consumption of caffeinated coffee does not significantly affect the generation of the SSVP signals in the parieto occipital region. Okay. Interestingly, frontal lobe activity in the brain was significantly increased, and this is what we found for the first time and reported that in the paper. Okay, the proposed method showed a good classification accuracy of more than 90%. Okay, so the limitation and the future direction over here, we had a very small number of participants that may have hampered the validation, and we intend to do the study on a higher population. Okay, and smaller electrode system, we used only 22 channel electrode system, uh, and now people are working on 64 channel EEG systems and all these things. So there is a lower spatiotemporal resolution over here. Hence, in future, if we are having a higher channel EEG system, then we can go for a study with the higher electrode system. Okay, and also long-term effects and the dose-dependent studies, they can be performed, and also the effects on different stimuli like ERP, evoked response potential, that may be visual or that may be the auditory one, that may be conducted. P300, then hybrid systems can also be conducted in future. So conclusion, if we conclude all these things, we find that these studies were focused on tracking the changes in the human electrical physiology of heart and cognition after the ingestion of the caffeinated coffee. The results infers alteration in the ANS activity, cardiac physiology, as well as brain cognitive activities after having this delectable beverage. However, for further validation, the study will be extended with a larger population size.